No, we have to set it down in the corrected form.
Okay, that's better. We good, Kathy?
Today you did.
Okay. Hi, everyone. I'm Shelley Tabb. I'm the library assistant here at the UAS Ketchikan Campus Library. Thank you all for joining us. I see some familiar faces and maybe some new faces too. This is our first Ask presentation of the semester, and I am really excited that Dr. Matthew Pawlus, our assistant professor of science, agreed to talk for us. He has his PhD in molecular biology from the University of Colorado. His main focus for his PhD was on kidney and breast cancer and how low oxygen levels or hypoxia drives cancer growth. So in just a moment, we'll welcome him with a round of applause. First, if you have your cell phones, can you silence them, please? And if you have a bag of chips, open them now. And I just want to remind you that your First City Libraries card works here. So if you have a library card and you've checked out at the public library, you can check out here as well. And up at the front desk, we have our email list. So if you're not on that and want to be, you can sign up before you leave. And also on the circulation counter, we have our AskUAS spring lineup, so you can mark your calendars for upcoming events. So I'm really glad that you all could make it, and let's welcome Matthew Pawlus.
Thanks, Shelley. Awesome. Thank you all for coming out tonight, too. It's great to have some people to talk to about some interesting stuff. So hopefully you find it all as interesting as I do. We'll see. Yes, you can see the title of my talk here is Welcome to the RNA World. I'm going to start just sort of really general, and then we'll get into the fun molecular biology of this topic. But I wanted to point out that just about every culture in the world has an origin story, so how the Earth and life first came to be around. Good thing I do too. Yeah, and the reason this is up for debate. In the 1700s, there was a philosopher, David Hume, who thought that this was probably because the human mind is sort of hardwired to view things as cause and effect. And we look around us every day and we see a lot of effects, and the causes aren't always apparent. So we sort of create these stories to sort of fill in the blanks. It's better to have something to believe in than nothing. What I'm going to tell you today is another story. It's another creation story about how life first appeared on Earth. It's a story, and this is kind of what we do in science. We pose stories when we don't have an idea of how something works. A little bit different than people did back in the day. At least we try to base these things on empirical data, and then we test them to see if they're true or not. So what I'm going to show you today is a origin of life story with some scientific backing to it. All right, so somebody said, science is a cemetery of dead ideas, even though life may issue from them, which I think is a pretty cool quote. I think they were actually commenting on the scientific method and how it works. So like I said, we pose these ideas, we test them. Sometimes they're shot down by our tests, which is okay. Then we come up with new ideas and test them again. And so from this, we end up with truth emerging, which is the goal. I also like this quote because some of the things we're talking about today involve, of course, science, but also this idea of life and death. So what is life? It's another fun picture of this creature here. No matter how weird life gets, and there is weird life forms out there, as humans, we're usually pretty good at telling what's alive and what's not just by looking at it. And there's a lot of different variability you can see in nature. Maybe not this guy yet, but who knows in the future. So let's play a game I like to play called Alive or Dead. We all look at this little girl, and alive or dead? Alive. Pretty obvious she's alive. It's not a trick question, it's not an old picture or anything. How about this? We got a crystal here.
Live. Dead.
Our current definition would classify a crystal as being dead. Sorry if that disappoints you. A fish here, and depends how long he's been out of the water here, but pretending he's a fresh catch, let's say he's alive. How about this one? Yeah, hopefully we didn't catch anybody there. There's a rock, and here we got a tree.
Alive.
It's obviously alive too. Jelly beans? Dead, hopefully. Yeah, so this illustrates an important concept in the way humans approach the world. In most cases, this is a perfectly fine way to get through your day. So most of the stuff we deal with on a daily basis falls into one of these two categories pretty easily. So we tend to group things that are alive into one category. So things like animals, plants, bacteria, fish, all those things. And they're mutually exclusive to things in the dead category. In the dead category, we put things like rocks, water, chemicals, air. And of course, we know that living things can die over time. That's usually what happens. We don't usually see the opposite happening. It's much harder to go from dead to something being alive, unless, of course, we're talking about zombies. But that was a previous presentation. So let's go back to this little girl here, and let's look at this in a little more depth. So we said she was alive, which is great. She looks nice and happy. Now let's pretend we take this little girl and we remove her brain. And we gently remove her brain. We're not monsters, we're biologists here. We take the brain out really carefully. We hook it up to a bunch of circulatory machines that provide blood and everything the brain needs. That brain would still be technically considered to be alive. It's living tissue. It's actually very hard to do that for very long periods of time outside the body, which is why if you're getting an organ transplant, you want to have fresh organs. If we were to zoom in on that brain a little bit, we'd find out it's made up of cells in there, right? And cells look something like this. This one's been prepared in a cool way to make it these bright colors, but all the tissues in your body are going to have cells in them. And cells, by our current definitions, are also considered to be alive. So our cell is alive, brain is alive, the girl is alive. But let's zoom in a little bit further. Inside the cell, you've got a bunch of other things. In essence, these are little organs inside the cell. They do little jobs. So here's a cell. This little organelle is called a mitochondria. Its main job is making energy inside your cell. Yeah, is this thing considered alive or dead? Any opinions?
Alive.
Right, so mitochondria exist inside of our cells, but by our current definitions, mitochondria are dead.
Oh.
And if we zoom in even further into our cell, we'll find things called molecules. So we have things like DNA, RNA, some proteins floating around, some RNA protein mixes. These things are also considered to be dead, even though that these things are required for life to exist, and they support the life of the cell, which is alive. They are dead by our definitions. So when you look at a cell is alive, but it's made up of dead stuff, because everything inside of it is considered to be dead. And therefore, by extension, the brain and the little girl are alive, but they're made up of dead stuff. And that's because this dead stuff, these molecules and organelles inside of our cells, they don't conform to this common definition of life that we typically use. So there's different versions of what this definition of what's alive and what's not. It sort of varies by your textbook. But in general, there's usually a few different criteria something has to fulfill to be considered alive. And it includes things like growth and development, so it changes over time. It's able to reproduce, pass on traits in some sort of way. It's able to regulate its interior conditions separately from the exterior conditions. Has some sort of chemistry going on inside of it that allows it to extract energy from the environment. And big one here, it is cellular. So current definitions of life say cellular life is the only type of life. All life is made up of cells. So what's the difference between living stuff and non-living stuff? Well, if you look at it close enough, you realize there really isn't any difference. So there's no difference between the carbon atoms that are in a diamond, something we'd consider to be dead, versus the carbon atoms that are inside your body, which we consider to be alive. We really just put emphasis on things that are alive because that's what we are, and it's useful for us to know what things are alive because those are usually things we can mate with, or we can eat, or things that want to eat us, evolutionarily. But really, this sort of a question of the difference between living and dead matter is really just a matter of perspective. So I'm a molecular biologist by training, and the term molecular biology is almost an oxymoron because biologists study living things, and molecules, like I just told you, are dead. So we sort of study these dead molecules that make things alive. And so I view life as sort of more like this. Instead of being two distinct categories, I see it as sort of this gradient between life and death. And I like to study things that fall right in the middle, that have some attributes of both of them. So of course, zombies are in there, somewhere between life and death, but they're not the only things. So Molecules like this guy, RNA, DNA, proteins, those things are considered to be dead, but they also have a lot of attributes that are similar to living things. And like I said before, they're required for life to exist. However, by our current standards, we consider them to be dead. All right, so this talk is going to be about a certain chemical that luckily for us doesn't get hung up on definitions, especially about life and death. It just did its own thing, and luckily for us, that actually ended up being great. So we're going to get into that in a second. This molecule, besides sort of doing its own thing, what that thing was, is actually going from life or from death to life. So this molecule, like I said, is considered to be dead because it's acellular. It's just a piece of, it's actually a bunch of atoms sticking together. But as we're going to see in here, it has a lot of characteristics we consider or we associate with living things, and it potentially was the impetus that started cellular life on our planet and gave rise to all the things we consider to be alive from these dead things in primordial Earth. All right, so yeah, these are big claims, and it's not lost on me that I'm just throwing this out there. So like Spock would say, where's the empirical evidence for this stuff? And that's what we're going to get into. But first we need to get a little bit of background, a brief history of all the life on Earth. So very brief in here. You can see in this diagram, which is possibly the most overused diagram in science presentations. I've seen it used in geology presentations, TED Talks on biology and evolution. But yeah, it's actually a fairly nice diagram up here because it gives you an appreciation of not only the scale of time that we're going to be talking about. Earth is currently thought to have been formed somewhere around 4.5 billion years ago. In the first early years of Earth, there wasn't much living stuff around. You don't see anything in the fossil record from these early time periods. And then as time goes on, at some point you start to see the very first life forms appearing in the fossil record. Although these things don't really fossilize very well, so usually you don't start seeing fossils until later on in Earth's history when you start seeing larger and larger creatures showing up. But here's us at the very top, most recent additions to this pile of geology beneath us. So our life history sort of extends through all these layers back a long ways in Earth's history. All right, so I want you to think about how you got here tonight. And some of you may have driven, I don't know if the bus runs out here, but you might have taken the bus, maybe an Uber, maybe you decided to put on a headlamp and run here tonight. Maybe you biked here. All those are great options. But regardless of how you got here, you use some sort of adaptations to get here, right? So different adaptations allow you to do things. And adaptations are just special features. Some of these features are shared across different types of organisms. Some of them are unique to humans. But here's just a couple of the ones you may have employed to make it here tonight. So vision, opposable thumbs, muscle control, breathing, memory, even technology can be thought of as an adaptation to humans that humans have. But all those things are designed to help you survive and to do what you need to do. If you happen to run or walk here, here's just a list of some of the adaptations that allowed that walking or running to happen. So a lot of body structure went into this, shapes of your heels, shapes of your legs, the way you metabolize energy in your diet, even the way areas of your brain are wired to keep you balanced and on your feet. Gist of it is there's a lot of adaptations going into even simple things like walking or running. And if you consider all the things we're able to do, we do more than just walk and run. All of those things are going to involve a ton of different adaptations or special features. So how did you end up with these adaptations? Well, if you took a biology class, you may have guessed it. Evolution is usually the answer if you don't know in a biology class. And evolution is really just how you end up with traits over time. So it's gaining more traits, losing some traits over time. So it's thought of as the frequency of traits changing within a population over multiple generations. And you really only need 3 things for evolution to happen. It's not that complicated of a process to actually get going. You got to have a genetically diverse population. So in our example up here, we've got some beetles. And the normal beetle up here is green, but every so often we get a mutant in here who's just got a slightly different DNA sequence, which makes him be orange. So these are all the same species. They just have variability. So you look around the room, or maybe don't look around the room if you don't want to see genetic variability. But you notice we're all humans, but we all look slightly different. So we have different facial features, different color eyes, colored hair, we're different heights. All these things are encoded in our DNA, and so we have genetic diversity as a population of humans. These beetles do as well, and most successfully evolving populations do. So you start at that point. The next thing you need is what's called selection pressure. So a selection pressure is easy to come by in the natural world. It's called natural selection if it's nature selecting for it. In this case, we have a bunch of crows move into this area of beetles, and these crows preferentially love munching on the green beetles. Maybe they taste better than the orange ones, who knows. But they eat the green beetles, they leave the orange ones alone. So that's a selection pressure. We're selecting against the green beetles, we're selecting for the orange beetles, which are increasing in their frequency in this population. And if this goes on long enough, eventually less and less green beetles make it to reproductive maturity. They don't pass on their genes for green color to the next generation. And so you see less and less green colored beetles over time, and more orange colored beetles because they're mating more, they're making more offspring. So that's differential reproductive success between those traits. And what you have there is basically evolution in a nutshell. So this happens over time. You end up with more and more adaptations accumulating in different frequencies over generations. You can do a similar thing with fruit flies, and like I said, humans as well if you want to get real masochistic with them. But take a bunch of humans or fruit flies, lock them up in different conditions, and expose them to different selection pressures. In this case, the fruit flies, some of them were put in a box with starch to eat, some of them with some maltose to eat. The ones that can eat starch are perfectly happy. They pass on their genes for starch digestion to the next generation. The ones that can't die. Same thing's true of the maltose ones, but with maltose. So over many generations, in the fruit fly box up here, you end up with a box of fruit flies that all can digest starch. In a box down here, you have a bunch of fruit flies that can all digest maltose, or most of them can digest maltose. So if you let this go on for more and more generations, they're going to start accumulating different adaptations between these boxes. And they're going to start looking more and more different. And at some point, they'll actually accumulate enough adaptations where they may not be able to mate with each other anymore. So think of Chihuahuas and Great Danes, right? They're dogs, but they've been adapted in different ways, and some of them are very small, some of them are very large. It's very hard, I imagine, to try to mate a Chihuahua and a Great Dane together, just due to the mechanics of the situation, because they evolve different traits. And so over time, you end up losing the ability to reproduce with each other between those populations, and those would be the start of two different species. So this is an event called speciation. And biologists like to keep track of this type of relationship with what are called phylogenetic trees. And phylogenetic trees sort of tell you how closely related, based on DNA sequence, you are to other organisms. So you can compare how related or how much DNA similarity you have between different species over here based on where they fall in the chart. And as you move to the left, this goes further and further back in time. Everywhere you see a divergence, like at these nodes here, this is symbolizing one of those speciation events. So you started with one population over here, and then you had two different selection pressures that split that population up, and they both ended up evolving different sets of adaptations over time. So if you look at this, the numbers here are in millions of years ago. So 90 million years ago, mice and humans had a common ancestor. 310 million years ago, humans and chickens had a common ancestor. And this goes all the way back further and further in time. And of course, besides giving us useful information, like who's more closely related to who, scientists also like to use these things to answer important questions, like which came first, the chicken or the egg? And as you can see here, the ability to produce eggs evolved long time before chickens actually appeared in the record, so now you know. And of course, we only had a few species on that last one, but you can make these trees as big as you want, or as big as you have evidence for. This one right here actually isn't the biggest. I just like it because you can still see some cool pictures in there. But they have ones now that encompass millions of species at the same time. And one thing you'll notice is, so out here on the very outer edge, this is current day, and as you move towards the center here, you're going back further and further in time. But we have all these organisms represented out here at present day. And as you work back in history, you end up with fewer and fewer organisms that we have evidence for. And they get sort of simpler and simpler in their adaptations. And this makes sense. So you tend to get more and more adaptations over time as you're exposed to more and more different selection pressure. So you're going to end up with more and more complex life. Now for some examples on here, we see also that the mammals all cluster together. So the very first mammals are going to probably appear on Earth somewhere back here 250-ish million years ago. Those are what we consider to be defined as mammals. Of course, they had ancestors that went back further, but they don't really take off and flourish until right around when the dinosaurs go extinct. Right here, you see their lines terminate about 65 million years ago. Then we start getting all these branches of mammals, different species evolving out of that. But the point being, the further you go back in time, the further or the closer and closer you get to a common ancestor. And if you go back far enough, you'll see back here, 2 billion years ago, we see plants starting to diverge off. Before that, all the creatures on Earth are classified as bacteria. And if you go back further and further, right around 3.8 billion years ago, you converge at one single point, which represents a theoretical common ancestor, the very first life form that gave rise to all of these things, which is cool. So all the life we have today is thought to be just sort of accumulating different traits through different selection pressures that started with that very first organism. Maybe right about there. So we have a name for this creature. Scientists call it Luca, and this again is hypothetical creature. We don't actually have fossil evidence for it. We just hypothesize that this would be around this time. But surprisingly, we actually can deduce a lot about what it probably was like. Again, it lived about 3.8 billion years ago. It may not actually have been the very first cellular organism on the planet, but it's the only one that gave rise to all the life forms we see today. So there could have been other earlier forms, but they just died off before they evolved. Okay, so what do we know about Luca? Well, surprisingly, more than you'd think. If you look at all things that are alive today, they share some common features. So they're all made of cells, is what, by our current definition, how we classify it, but they also all have DNA, RNA, proteins, and a cell membrane. So it's likely that we all have these things because Luca had these things as well. So even bacteria have these things too. So it's not a big leap to assume that these things all were already encoded in our DNA from Luca. Now we also assume that this was a single-celled with a circular chromosome, just because we know that's what bacteria looked like. And bacteria were sort of the first creatures to evolve. They're still around today, and that's what they look like. They're one cell, they have a circular chromosome. So why cells? Well, cells are a good deal if you want to keep alive, which is why they're so popular on the planet. So they're really packets of biochemistry. You have this membrane, and inside that membrane you can have different chemistry going on than outside that membrane. So cell membranes sort of divide reactions up between the inside and the outside. They allow different things to happen on either side of that membrane. DNA, also a great thing to have around, which is why we all have it. We all know DNA plays an important role in storing information in our bodies. So this is what DNA looks like. The information we're talking about being stored up is actually the information used to build proteins in our body. And that information, the language it's written in, is the sequence of these specific chemicals in the middle of DNA called base pairs. So they come in 4 different flavors, A's, T's, C's, and G's. And that sequence in your DNA basically determines what type of protein can be made from that piece of DNA. All right, so DNA is great. It's a pretty stable molecule. It also allows us to pass on this information to subsequent generations, which is why we have it. Now, RNA is a molecule you may not be as familiar with as cells and DNA, but it's important nonetheless, and all living things have it. So RNA is actually an important step in what's sometimes called the central dogma of biology in textbooks. But this basically states information flows from DNA into a molecule of RNA, and then ultimately ends up as a protein. In your cell looks something like this. Your DNA lives inside your nucleus, which is this chamber here. DNA is really big, and proteins end up being made out here in the cytoplasm. So DNA can't just walk from the nucleus to the cytoplasm. It can't fit out of these little pores that allow it passage. So to get around this, you have a little piece of mRNA, or RNA, that carries this information. It basically copies down information in your DNA and carries it out to the protein-making machinery in the cytoplasm. Now, it turns out RNA has a lot of other functions as well, but it's still important for this function for us. And you also find RNA in bacteria which don't have nuclei. So this also suggests that RNA also is doing something else in them as well. We'll talk more about what that is in a second. Okay, so we know a little bit about what Luca looks like, maybe why it has some of the adaptations it had. why it was so successful at producing all of the life we see today. But what about before Luca? This is really where we want to concentrate our efforts during this talk. This is where our questions lie. But before Luca, there were no cells around. So we don't have anything alive according to our current definition. So this is prebiotic world. There's only dead stuff, nothing alive because nothing's made of cells. What was around, though, was chemicals. And chemicals, like I said, may have mediated this transition from dead stuff into the first living organisms. Okay, so how did this happen? Yeah, I won't say that people didn't have any ideas. There were lots of ideas floating around for long periods of time. Just none of them that really panned out or really had much evidence to support them. Until right around, well, so I guess for some of these examples, people thought maybe DNA had been around beforehand, but the problem with DNA is you need proteins in order to copy DNA, to synthesize it. So then people said maybe proteins came first, but proteins are encoded by your DNA, so you had to have DNA. So it was this chicken and the egg situation. And then somebody in the 80s said, what about RNA? They probably weren't the first person to think about this, but they were the first person to sort of put this down in a formal hypothesis. But RNA is actually more than just a messenger between the nucleus and the protein building machinery in cells. It actually can do a lot more stuff. And to put it in other words, I don't know if you guys are into memes, but this basically summarizes it. But as it turns out, RNA is extremely simple chemically, meaning that in experiments in the lab, you can actually synthesize this stuff just by mixing the right chemicals together. And these chemicals are ones that have been shown to exist on primordial Earth. So in theory, RNA could have formed just through self-assembly. If you had the right chemicals in a shallow ocean somewhere, they mix around, you could have ended up with probably a shorter version of this molecule, but nonetheless, you could have ended up with some RNA showing up. So RNA, potentially, before Luca, was maybe the dominant form of chemical life on the planet before cells took off. And this is the hypothesis that we now call RNA world. So RNA world refers to this time period before cellular life emerged, but which ultimately made this transition from chemical life into the cellular world we see around us today. All right, so we don't know all the details of RNA world yet. We can't figure out exactly the sequence of events that led up to it. But we know that something that we consider to not be alive must have been involved because, yeah, we're here. So if we weren't here, we never, so there were no cells around to basically create life. So we wouldn't be here unless something dead at some point in time became alive and created cells. So the question is, how did this happen? Was it magic, random processes? This is usually where somebody shouts aliens. Yeah. It just pushes the problem on further. It doesn't solve your problem because aliens are alive. So at some point in their evolutionary past, someone would have figured this out as well, but it should have come from something dead, must have transitioned to life as well. I'll argue here, and the RNA world hypothesis supports this, that this is actually a transition that could naturally happen between death and life just due to normal biological processes. Okay, is there any evidence that dead stuff could come to life in more recent biology? So currently science says no. This is Louis Pasteur. He's a famous microbiologist. He's famous for inventing the pasteurization process that sterilizes all your beverages and milk and stuff today. But he famously proved, so back in the day when he lived, people noticed that when they left pieces of meat just laying on their countertop for extended periods of time, stuff would start growing in that meat. They'd see mold and bacteria and maggots and things. So it led people to wonder, is life just something that could spontaneously show up? Is it just magically appear when you leave dead meat laying around? And Louis Pasteur said, well, let's test this out. And he basically showed that if you sterilized your meat sample or your broth, which was nutrient rich, and kept it in a nice little flask that prevented things like air and spores from blowing in there and taking root, that you never got new life forms forming. So this gave rise to the cell theory I mentioned earlier, and this is the theory that states all living things are made of cells, and you can only get new cells formed from previously existing cells. So that's where science stood for the longest time, before RNA world, which is why we consider cells the most basic unit of life, because you have to be a cell to be alive. But, like I said, this has had to have happened at some point in our past, because we are here, and we're having this talk. So we know that life hasn't been around forever on planet Earth. It had to have come from somewhere, and the fact that all we had before cellular life emerged on the planet was chemicals laying around, it begs the question of, are chemicals alive in some sort of way? I wanted to point this out, that people have done experiments to show that other biological chemicals could have existed at the same time as pre-Luca life, or this RNA world hypothesis was going on. So people have done experiments to show that if you mimic conditions of primordial Earth, you can actually get the formation of simple amino acids and things like that, and other chemicals that are around. So we've got this rich soup of chemicals being created and falling apart and doing chemistry with each other, for generations and years before we end up with the first cellular life form. So the question is, are chemicals alive as well? Or do we just have a bad definition for life on Earth? Since we are cellular life forms, we only extend that sort of award of life to other things that are also made of cells. And this has always bothered me, but if you watch, not just the makeup on some of these, but in Hollywood movies, the way that alien life is portrayed is actually pretty boring, because it's so similar to human life, right? Like, we have similar body plans here. Everybody's looking sort of hominid. They're bipedal. They have forward-facing eyes. Basically, all these analogous features that are only useful if your species has evolved under the same or similar selection pressures as humans have, and if your planet is pretty much pretty similar to Earth as well. So this has always disappointed me. And yeah, I find it more interesting to think about the different possibilities for life out there, especially if you consider that life could exist in the uncellular form, or this abiotic form that we're calling now. But if there's life out there in a chemical form, it could take all sorts of interesting morphologies. So maybe this rainbow film that could be dancing across a puddle might have evolved into intelligent life on another distant world somewhere. I think that would make a pretty cool movie and a cool depiction of aliens. And yeah, I'm not the only one thinking this. So NASA has also realized that there's certain shortcomings with a current biological definition of life, mainly that it's too narrow in focus. So while they're extending probes into the universe looking for life on things like comets and asteroids and even planets in the near future, they came up with a sort of broader definition for how they would identify life when they encountered it in a possibly alien environment. And you can see here, it doesn't mention cells anywhere. They say a self-sustaining chemical system that's capable of Darwinian evolution. So basically chemicals that can evolve, which is pretty cool to think about. So what would that actually look like if you found it? And I made this little list to sort of make some important bullet points of things that you should look for if you're ever exploring an asteroid somewhere looking for signs of life. You'd want to have a chemical that could store information in some way. It would have to also be able to pass that information on if it wants to evolve, which is one of the requirements. It would have to be self-replicating too, so it'd have to be able to replicate itself, and it'd have to have a metabolism. So it'd have to do some sort of chemistry and interact with its environment in some way. So if this definition is good enough for NASA today, why not apply it to Earth 3.84 billion years ago, before there were cells around? If we were to use this, and if there were astronauts using NASA's definition around back 4 billion years ago and they came to Earth, would they have found RNA? And according to this definition, would they have classified it as the first living creature on our planet? Let's talk a little bit about RNA and why maybe the answer is yes to that question. So here's RNA over here. We've showed a couple pictures of it. You noticed it kind of looks like DNA. Yeah, chemically it is a lot like DNA. So there's a couple of big differences. Both DNA and RNA are what are called nucleic acids, but they're all made of sort of this common combination of three different chemicals. You have a phosphate, you have a sugar, and you have a nitrogen-containing base, which is all these rungs on the ladder. These are those things whose sequence makes up your genes that encodes for proteins. So RNA has those as well. It just uses a slightly different sugar. So it uses a sugar called ribose instead of deoxyribose, which DNA uses. It is also single-stranded, where DNA is double-stranded. And it has one different nitrogen-containing base. So out of the four bases that DNA has, RNA has one that is different. So pretty similar stuff. And because of the similarity, mainly that it has these nitrogen-containing bases, RNA can hold information possibly just as well as DNA can. So it has a sequence of 4 different nucleotides. And again, this is what we're talking about when we're talking about information in DNA. It's the sequence of these chemicals in the middle of the ladder. So the fact that this goes CAGC and not CATC means that this gene is possibly going to code for a completely different protein that would be used for something different in your body. So DNA holds that information. RNA does as well. Like I said, it uses the same bases. It substitutes uracil down here for thiamine, but you can see they look pretty similar as well. But it can hold information in that sequence. What's more is that RNA can participate in metabolism. So because it is single-stranded, unlike DNA, it can actually loop back around and form connections with itself. So it can dimerize with itself through those base pairs. They'll start to stick together, and it can create all kinds of really cool structures. And here's some examples down here of different structures that RNA can fold into. Now, because it can fold into these structures, this allows it to have certain enzymatic functions. So it allows it to do chemistry where it doesn't destroy itself. So chemistry where it's not just breaking itself apart and falling apart or being consumed by the reaction. Whenever you have something that does that, does chemistry without being destroyed, it's what's called a catalyst. So we say that RNA can actually catalyze chemical reactions because it can form these cool secondary structures. And here's a strand of RNA right here. It's linear. This one's actually pretty cool because it can fold two different ways. And it can take on two different forms. And because of these forms, it can have multiple enzymatic functions. So we've already seen in these first two examples that RNA can behave sometimes like DNA in that it stores up information, but also sometimes like proteins, which are typically the catalysts we have around in our body today. We use them to catalyze our reactions for the most part. RNA is also self-replicating. So I said earlier that DNA, in order to replicate, it requires proteins. But RNA, because it can form these catalytic structures, it can actually catalyze its own self-replication. So a strand of RNA can fold into a certain structure that allows it to make copies of itself over and over again, which is one of those qualifications for something that's alive. And so, of course, if it can replicate itself and it can hold information, and if you think about this, if it's self-replicating, it's passing on this information to every subsequent generation. If that DNA, or the information in that RNA is changed between generations, that is, in essence, mutation. And so over time and over generations, you get selection pressures acting on these RNA molecules, selecting for very specific mutations and very specific enzymatic abilities in RNA. And over time, RNA can evolve into more and more complex structures. So again, here's our definition of evolution we saw earlier. Frequencies of traits changing within a population over generations. The only difference between RNA and animals, or cellular stuff we were talking about earlier, is that our population is RNA here, and it's not cellular based. So, do we fulfill the three requirements for evolution to happen, though? Number one, we need a genetically diverse population. So, does this arise in RNA? And the answer is yes. So the genetically diverse population of RNA is just RNA molecules that have different sequences of those bases. They might have a limitation on which bases they can incorporate into themselves at any given point in time. They might sustain mutation, just like DNA does today. Maybe they're exposed to some too much UV radiation shining down on their pond that they're floating around in. But however it happens, differences in DNA sequence are going to, RNA sequence are going to happen. And because that sequence is different, that actually changes the ability of these guys to fold into different structures. So different sequenced RNAs fold into different shaped RNAs. Those different shaped RNAs then are able to catalyze different types of chemical reactions. All right, so for example, we're going to follow the evolution of these three RNA molecules over time here. So first off, genetic diversity. So it exists here. I'm just choosing a section of just 4 nucleotides in these 3 molecules to show you that they're different. And they're not different by that much. So there's an A here, a C here in the first position, and over here you have a U in the last position and a G over here. So they're only differing by 1 or 2 base pairs. Okay, so genetic diversity, it's there. How about selection pressure? What would that look like for RNA? So selection pressure is something that's going to preferentially select for the survival or reproduction of one of these species over the others. And let's pretend, well, we don't even have to pretend, but these guys, if they want to grow or replicate, reproduce themselves, they're going to need the raw materials to build a new RNA strand, like the nucleotides, like those information-containing rungs of the ladder that make them up, those A's, C's, U's, and G's inside of here. So these things will be floating around in their pond. Hopefully they're all there. As long as all of them are there, all 3 of these RNA molecules can replicate themselves and survive equally. But if we run out of one of these nucleotides, maybe there's a competing RNA strand across the pond that's eating up all the A's from our pool over here. Or maybe, just due to random circumstances, we don't have access to A's anymore. Now all of a sudden we've got a selection pressure. So we can't make any of the sequences that have A's or require A's for their reproduction. So these 2 are not able to successfully reproduce themselves. The one in the middle, though, which doesn't have to worry about A's in their environment because it doesn't have any in its code, it's perfectly capable of replicating itself over and over. So that is what we would call differential reproductive success. So after several rounds, we're going to end up with a population that's mostly these guys in the middle. We might still see these other species in our RNA population, but they're going to be less abundant by a long shot compared to these ones over here. So we've got genetically diverse population, selection pressure capable of influencing these guys, the evolution of RNA molecules, and then differential reproductive success, which is all you need for evolution to really happen. Of course, you need time in order to evolve more and more complex structures. So every round of replication, every mutation sustained in this RNA molecule is going to be passed on and selected upon by natural selection over many, many generations for many years. But ultimately, you end up with more and more complex adaptations emerging that allow molecules to have a better survivability, better reproductive efficiency in their environment, so a better fit for the environment. And something like this can ultimately evolve into something like this. And this is actually a ribosome, ribosomal RNA that you find in your cells today. It's a bunch of RNA floating around. Okay, so besides evolution, that's great. These molecules can evolve. They might be able to change their abundance and their adaptations. But what about making that jump into cellular life? They need to have some other adaptations, right? So as it turns out, like I said before, having a plasma membrane or a cell is a big evolutionary step forward because it actually enhances your ability to evolve over time. So let me explain what I mean here. Containment inside of a semi-permeable membrane or a bubble structure here, that's in essence what a cell is. But if you can contain a population of RNA in here, what you have is an isolated reproductive system. So in this reproductive system here, you can get genetic material being exchanged between these RNA molecules. You also have control over the chemical conditions inside this membrane. So you can isolate or pull in chemicals from the outside. So food particles may be able to pass into this membrane where you can use them for replication of your nucleic acids. But other damaging things you don't want in there, things that might destroy you, like other enzymes or other RNA molecules, can't access your population. So it's like a little safe haven, a little island for these RNA molecules if they can end up with some sort of plasma membrane around them. All right, so how do you get one of those plasma membranes? You don't just run down to the store, especially if you're an RNA molecule 4 billion years ago. But as it turns out, membranes aren't that hard to make. So if you've ever taken a mixture of oil and water, shaken it up in a bottle, you'll notice that it doesn't mix together. You'll also notice that some of the oil sometimes forms little balls or little droplets that sort of float around in the oil or sort of float around in the water and bob around. You're pretty close at that point to making a primitive cell or primitive plasma membrane. So this is what, in effect, a plasma membrane looks like. It's a hollowed out ball, and you can form these spontaneously just by throwing in a special type of lipid. Oil is a type of lipid. Things like fats are also lipids and waxes. But since these things don't really like to mix well with water, they tend to just clump up together with themselves, but they'll spontaneously form a little hollowed out ball if you throw the right ones into water. And that's a perfectly acceptable semi-permeable membrane that would be perfect for RNA to set up a little family inside of and evolve over time. And this little artist rendering here shows you what this might look like. You can see a plasma membrane forming here, made up of a bunch of lipids. And inside there you have some RNA floating around. So, of course, this could obviously lead to the very first cellular life. You've got nucleic acid inside of an aqueous environment in here that's doing some sort of chemistry. It's replicating itself in here, and on the outside it's encompassed by this plasma membrane. So it's already starting to look a little bit like a cell. And what I wanted to show you was this little couple little clips from this TED Talk. It's a really cool talk. You can check it out to see a little bit more about chemical evolution and some cool experiments in there. But this guy's name is Martin Hansik, and he's in his lab in Denmark. They study these sort of protocells is what we call them, but they're these, in essence, oil droplets. Inside the oil droplets, he'll put some sort of chemicals that do chemistry, and he shoots these little videos, and surprisingly, they behave a lot like living creatures. So let me see if I can, I'm just going to fast forward to the parts that are relevant here. We'll start right around here. So what you're going to see is a petri dish with the water in there. There's also some chemicals, and in there is his protocell. So it's an oil droplet, and inside that oil droplet, there's some chemicals doing some chemistry. And as you can see, it actually has some motility. As long as there's energy flowing through the system, the thing will move around. And as he adds this in here, this is, in essence, the food for the cell. This is the reactants that the chemical reactions inside the protocell need in order to maintain their chemistry. And so it will actually be attracted to and seek out the highest available concentration of that food source, or the chemicals it needs to complete its chemistry, which looks a lot like a cell looking for food. Let's see, there's another clip here. And he also, here's another setup with these protocells in here. In this one, we have two different cells that we're starting off with, some orange ones and some blue ones. They're the same, they're just doing different chemistry. In some cases here, you'll actually see one of the blue cells is going to absorb one of the orange ones, which is something that may have happened in an RNA-based world when we started getting these plasma membranes. They might have fused, and then all of a sudden you have a bunch of more RNA molecules in your pool of reactions for evolution to take part in. As you can see there, this one actually did undergo this sort of spontaneous replication event as well, which is pretty cool. So at some point, if you increase the size of a cell too big, it makes sense for it energetically to split itself into smaller pieces. It's easier to maintain. And now those small pieces, they both have a mixture of the chemistry that was going on inside that parent one. So in essence, it's what we call a protocell, but it's one big step forward from non-living stuff towards something that looks alive. All right. So besides the plasma membrane, there's a couple other things we need before we're getting up to the very first cells. What about proteins? Where do those things come from? So yeah, proteins are awesome to have around. Evolutionarily, they do a lot of the stuff that we need to stay alive. So yeah, proteins, they're made up of building blocks called amino acids, and there's 20 different amino acids. So think of those as different shaped Legos. You can put them together in a lot of different ways because there's so many of them. And so you can create a lot of different diverse structures of proteins that do a lot of different jobs. So we use proteins for all sorts of things. There's structural proteins. We use proteins in our muscles. Antibodies are proteins, so they're part of our immune system. Proteins are used for cell-cell communication because they can take on all these different shapes. And so, yeah, it's a big evolutionary advantage if you can make a lot of different shaped molecules that do different jobs for you. Okay, so how did proteins show up, starting with RNA? Well, as it turns out, yeah, earlier experiments show that we could have had in this primordial Earth a sort of primitive version of some of these amino acids, so these building blocks of proteins floating around. Now the problem is that in most cases you can't stick these amino acids together unless you have some sort of catalyst to shove them together. And like I said earlier, RNA can act like a catalyst if it's formed up. So what we think happened is that an RNA molecule grabbed an amino acid, another RNA molecule grabbed another amino acid, they got together and they catalyzed the joining of those two amino acids. And they did this over and over to build a long chain of amino acids, which is a protein. And that, in essence, is how you would get proteins being built. And we see evidence for this. If you mix proteins or amino acids together with RNA molecules, RNA molecules will stick to amino acids. You can do it in the lab today. They actually have their favorites as well. So certain RNA molecules will actually preferentially stick to certain amino acids, which is also interesting to note. And then they can potentially catalyze the joining of those two amino acids together to make proteins, and they still do this today. So if you know anything about protein translation, you'll know that it actually involves 3 different types of RNA. You have messenger RNA, which is taking information from your DNA out to the machinery that builds the proteins. That machinery is actually largely made of RNA. This is ribosomal RNA, which makes an organelle called your ribosome. This is the machinery that actually catalyzes the joining of amino acids together. And this little guy up here is what's called tRNA or transfer RNA. He's the one that binds to individual amino acids. He brings them to the ribosome, and the ribosome joins them together. So this system could have evolved even before DNA on the planet. You could have had RNA giving rise to proteins. And then, of course, once you have proteins, you can use proteins to synthesize stuff like DNA, which is pretty fun. So yeah, let's talk about DNA. So we all have DNA. We talk about how great it is. It actually is a really awesome molecule. It's beautiful. It's in this nice double helix shape here. It contains all the information to build all the proteins you need in your body in this base here. Other bonus things about DNA that you may not have known, it's less reactive and more stable, therefore, in normal environmental conditions than RNA. So yeah, if you go out in the world and you start looking for RNA, it's harder to find RNA, but you might be able to find DNA from several thousand years ago if you look in the right spot. It sticks around for a while. It doesn't break down as easily as RNA, so you can keep your genetic information more stable for longer periods of time. It also is more resistant to some of the mutations and errors that come with RNA synthesis. So when RNA replicate themselves, they're not always the best at it. They can make mistakes. There's less mistakes when you replicate DNA. All right, and we've already seen a picture of DNA and RNA. We know that they're fairly similar. Only the big steps we have to do is change our sugar from ribose to deoxyribose. That's mainly where the stability comes from. So deoxyribose is much more stable than the ribose that we use in RNA. And this is great for storing up information for long periods of time and passing them on with a good amount of fidelity. The bad part is because you create this double-stranded structure, which also plays into the stability, you lose the ability of DNA in most cases to sort of loop back around on itself and form these complex three-dimensional structures that RNA does. So DNA doesn't usually have much catalytic activity. So DNA is only beneficial if you've got something else that can catalyze chemical reactions, like RNA or proteins. So we think RNA was probably the first thing to show up on the scene, and then it gave rise to probably proteins first, and then DNA a little bit later. All right, so all of these adaptations should sound familiar. Having DNA, having a cell membrane, proteins, RNA, these are all the things that we said were common to LUCA, that very first life form, and also things that we find common in all of our cells today. So back to this original definition of life. Now that we've evolved into LUCA, LUCA conforms to this definition, and so of course it's alive. It gets all the credit as being the first living organism, which then goes on to evolve and accumulate adaptations to create all other living life on the planet. So all of this stuff, starting with LUCA down here, we end up with populations becoming segregated, reproductively isolated, being exposed to different selection pressures, evolving different adaptations, and all of that goes into making all the different variety of life we see today. But they all still retain those commonalities we saw in LUCA that could have eventually, or could have evolved from this RNA world. So including all these things over here. And if you think about this, if you want to think about it this way, it's up to you, but if not, you don't have to. But really, all of this genetic diversity we have is this downstream event of RNA trying to replicate itself, trying to get more nucleotides to build a copy of itself. So really, we're just all sort of the products of RNA. And you can even extend this out besides looking at the physical adaptations. Of course, physical adaptations as well as genetics can play into a lot of other more complex behavior and adaptations. So things like cognition and emotions. These are all things that we have evolved as a species to interact with each other that promote our survival as a species. Those things wouldn't have evolved without RNA world to begin with. So think again about how you got here, whatever your means of transportation was, and all those adaptations. And again, sort of putting a sort of end cap on the RNA world and summarizing it up here in these three little panels. Think about RNA. So RNA, it predated cellular life. It was around. It dominated the planet for probably millions of years before the first cellular life form showed up. Cells themselves could have been an adaptation of RNA, just to help them do their chemistry, help them get the raw materials they need to reproduce. And from that, they could have evolved on to create proteins and DNA. And if you think about this further, all life today potentially exist because of RNA and those adaptations that had accumulated early on. So maybe multicellularity and all this complex body type and all those adaptations you used to get here, the Uber you called, all those things were possibly just the product of RNA trying to get its raw materials from the environment and facilitate its own reproduction. And all this, of course, is to promote the process of evolution or find the most fit RNA sequences around. So, yeah, so it's all due to RNA, and RNA is awesome. We're sort of out of RNA world at this point, so we've moved on to cells, and then we're now in a cell-based world. But RNA hasn't gone away altogether. We still have RNA around, and I've already mentioned a couple jobs it does, but it actually is very important in our current world and in current technology. So how does it impact us? Well, knowing about RNA world helps with how we might want to define life. So of course, while we're looking for life in different planets, it's stupid for us to assume that all life is going to be cellular based, when cells themselves are just an adaptation that could have arisen through evolution. So what we should be looking for is something a little more broad, maybe like NASA's definition. The definition of life should extend to things to include self-replicating chemicals. Our knowledge of RNA and its role in evolution and its current roles in maintaining cell physiology are also important for a field of biology called synthetic biology, where people try to create life forms themselves. So we start with the cell at this point. We basically remove everything except the cell membrane, and you can put in whatever you want in there. The idea is that you can design a DNA or RNA-based sequence that has a certain function, whatever you want it to be. Maybe you copy it from another organism. Maybe you make it up completely new. But you can get that DNA sequence synthesized online. You can buy it. I don't know if Amazon sells it, but you can get it from other websites online. They'll ship it to you. You can throw that into your cell membrane. And these cells, in essence, are a lot like the early protocells and maybe even like Luca. They're able to reproduce themselves, they do chemistry, and they do whatever job you want them to do. So people have already used synthetically developed organisms to do things like clean up oil spills or radioactive material, and they're developing all kinds of cool applications for them. RNA is also important in saving lives. So besides defining life and creating life, we can save life with RNA. It's the basis of a lot of molecular medicines that are either designed to treat diseases, including things like aging. So you can use actually an RNA-based enzyme. So one of those RNAs folded into those cool structures that catalyze reactions. You can use them to rebuild these structures on the ends of your chromosomes called telomeres. Telomeres shorten every time your cell divides. And when you get old, it's usually because you have a lot of senescent cells. It's because you basically have divided your cells enough where these telomeres have gotten too short. And when your cell senses that the telomeres are too short, it basically goes into sleep mode. It sits there, but it contributes to a lot of the phenotypes of aging. So if you can rebuild telomeres, maybe you can reverse aging, stay young forever. It's pretty cool. But that's RNA-based. It's an RNA-based enzyme that can rebuild those. It's interesting, we actually express that enzyme when we're young, but we lose this expression over time as we get older. And if you look at a lot of organisms that have extremely long lifespans or even ones that can regenerate, a lot of them will actually have this enzyme re-expressed in them. Anyway, maybe that's another talk. But you also use RNA in other forms of medicine, including gene therapy. RNA has been designed to be packaged inside of a virus. You infect somebody with this RNA-containing virus, but the RNA you put in there doesn't make them sick. It actually will seek out and destroy RNA from other viruses like HIV and Hep C and SARS and flu. So you can use it as a treatment to sort of counteract other viral infections. Another fairly common process used in the laboratory is one called selects, which in essence is a forced evolution of nucleic acids. But you start with a pool of DNA or RNA that's just random sequences, random structures. And through several rounds of this selects process, you can select for nucleic acids that bind to a certain molecule that you're interested in. So in essence, you can replace antibodies with these RNA-based molecules. And they're typically more selective than proteins as well, so they're better at binding to things. But people have used this method to make drugs that are based on RNA that target antibiotic-resistant bacteria or cancer genes as well. Okay, and maybe most importantly, RNA world, it sort of challenges the way that we not just define life, but how we think about life and death, right? So we typically like life and we think of death as this cold and unforgiving thing, but really, if there's no inherent differences between the matter of living and dead stuff, and if RNA world's true and dead stuff or stuff we consider to be dead can make this transition to life, then really what we're talking about is just sort of phases of matter. So they're just different states of matter, but inherently they're all equal. So maybe that changes how you look at life and death, maybe not. It just seems like an arbitrary distinction that we make. So what's our purpose on this planet? I included, the purpose of life is included in the title of the talk, so I don't want to shortchange you here. So if you're an RNA molecule, you may look at humans, and this might be how you define your purpose. So are humans just this sort of robot that RNA drives around, consuming resources to suit its own needs? Like I said, if you're an RNA molecule, maybe the answer is yes. I don't know, you can ask them sometime. But they would tell you that your purpose of being here is to obtain resources to do your chemistry, propagate diversity of your molecules, and then allow natural selection to select the best fit ones for the environment. So, and then pass those on to the next generation. That's kind of a boring purpose. I would challenge it with this. From the human perspective, and keep in mind we're not living in the RNA world anymore, we're in the cell-based world, and we are now the dominant forms of life on the planet, so I think it might be more valid to look at this from the human standpoint. But the purpose of life is really whatever you want it to be. So think about that huge evolutionary tree of life. Humans are at the very top, the outer edge right there. We've basically accumulated all these adaptations. We're the most adapted, the most fit creatures for the environment at this stage of life. And so you sort of earn this right to make your purpose whatever you want it to be. You're very fit for the environment, you're capable of a lot of stuff, so you can go out and do whatever you want for the most part. If you don't, you won't pass your genes on. So if what you do ends up killing you, you're sort of doing natural selection's job for it. So yeah, keep in mind the RNA world, it's an explanation of how life arose, but think of it like a distant ancestor. RNA world shouldn't influence your choices today any more than a distant ancestor might influence your choices. So of course, having said all that, everyone's got their own personal suggestions of how you should make your life purposeful. These are mine. Go out, do a lot of stuff, learn a lot of stuff, especially about the natural world around you. It's probably weirder than you think, and you'll enjoy it. Pass on what you know and your experiences to somebody who's willing to listen. Do talks like this. And also don't take your life too seriously. It's just a fad, like Mick Jagger said. And of course, be kind to your chemical friends. And who knows, one day they might end up ruling the world. So that is the end of my talk. So I'm sure there's questions, and I'll go ahead and answer them. Yeah.
How do the viruses fit into this?
Yeah, so they would fit in that gray area between life and death, right? So they have, if you look at a virus, what it is it's just basically a protein coat wrapped around some nucleic acid, either DNA or RNA inside of it.
So it can be both, DNA
Well, so people argue it. In most textbooks, it's classified as a non-living thing, but it does have a lot of attributes of living stuff. So viruses can change over time. They can evolve into different things in different forms. They can't reproduce on their own necessarily, so they need to infect a living cell. What they do is they hijack the cell's machinery that it uses to make copies, and they use the cell's machinery to make copies of viruses. So it can't reproduce on its own, but yeah, I think it would fit right in that gray area between life and death.
Would be long time after Luca or whatever that
as far as evolution of these things happen. So yeah, so viruses have been around for a long period of time. What you would need, obviously, I think RNA viruses would have preceded DNA viruses because RNA seems to have preceded DNA in all cases. But once you have that RNA around, like I showed, it can start binding to amino acids and catalyzing the formation of proteins from the amino acids. So I think viruses could have predated cellular life on here as well. But then you run into that problem of if they can't reproduce themselves without a cell, how are they going to stick around?
Yeah.
So yeah, I don't know for sure, but possibly it could have been before Luca. Yeah.
Yes. How related are systems biology and synthetic biology?
They're kind of two separate things. You can think of some applications maybe where they overlap, but systems biology it's I won't say it's a new approach, but it's definitely gotten a lot hotter of a topic more lately. You see a lot of institutes popping up for systems biology. But what systems biology is looking at sort of a more holistic approach to biology. So instead of in the past, you've typically had people like molecular biologists studying DNA, and then further down the road, you'd have somebody who is an expert studying I don't know, some sort of wildlife biology, but these sort of seemingly unrelated disciplines. And systems biology sort of seems to integrate these things. In most cases, you're not dealing with something, the field's that diverse. You're usually dealing with, like, in a systems biology institute, you have somebody who's like a virus expert, and there might be somebody who's a tumor expert, and they get together and they come up with a gene therapy virus that can target tumors or something.
Nice.
But yeah, synthetic biology is also emerging. It has a big role to play in industrial applications because of the whole bioremediation angle, so cleaning up our trash and things. But I think we're just sort of starting to get into the potential with synthetic biology because, like I said, you can program in essence life to do whatever job you want it to do. The only limitation we have right now is knowing that language, so knowing which genes can actually do what sort of functions we want, putting them into a system. But once they're in there, then that life form is basically able to replicate its own hardware. It goes on, it replicates itself indefinitely. So, yeah, obviously there's lots of issues with what would happen ecologically if these modified organisms get out, and we're sort of introducing a new species and all that. But yeah, those are, I think, inevitable problems that we're going to have to discuss at some point. But
And you also said that there are services available online where you can send in
Oh, yeah.
a sequence of, like a DNA sequence, then they'll send you.
yeah, so making DNA, it's actually, if you got a machine for it, the machine's really expensive, but to make DNA, it's really cheap. There was a video that talked about, so obviously I said that the building blocks of DNA are, you have a sugar, a phosphate, and these nucleotides, the nitrogen-containing bases inside of them. So there's only these three parts, and most of them are pretty cheap to get. So it was something like $25 of sugar gives you enough DNA to, you could print with a DNA machine everybody's genome on the planet. And it gets cheaper the more people use it. So it's a fraction of a cent per genome or per nucleotide that you put in there. But yeah, so you would write your ACT and G sequence that you wanted online. Like I said, you can copy and paste it from websites. So there's a good database. NCBI maintains a great database of different sequences of genes from different animals. So if you wanted to, and people have done this, you could go in and find the gene that codes for jellyfish fluorescence, the protein. And you could copy that gene sequence, paste it into your order on IDT, a company that can give you your whatever oligos you want, but they'll print you a DNA. But then, yeah, they get the sequence, they plug it into their machine, and it prints off a little bit of DNA for you. They put it in a tube, and then they mail it to you, and then you got it. So then you can use it for whatever you want. Yeah, in the back.
So I don't know if this is covered or if it's outside of the realm of the discussion, but I'm a little confused at how we go from a table of periodic elements that I guess we use, they're inorganic, they're all dead, to RNA.
Yeah, so there is a whole, so we talk about RNA world today, which is that section of time before Luca, but there's a whole nother history before RNA that would have led up to the events of RNA. Because RNA is pretty simple, but it still is going to involve a little bit of chemistry there for that to happen, right? So there's a hypothesis that called PAH world, which is polyaromatic hydrocarbon world, which people think might have predated RNA world. And this is basically, polyaromatic hydrocarbons are these like multi-ringed structures made of carbon rings, right? And they're actually fairly common in the universe. You find them a lot on like comets and the moon and all these, they're kind of almost everywhere. So the idea would be is in some way these polyaromatic hydrocarbons ended up on Earth before RNA world. They were the ones that were floating around, and ultimately, those ones mixed around maybe with some other chemicals that were just sort of around in Earth's ocean or on its crust somewhere, and those are the ones that did chemistry that ultimately led to RNA. But the idea might be that, possibly you could look at those polyaromatic hydrocarbons as evolving through a process as well. So they would have taken on a bunch of different shapes initially, and only the ones that would have been successful and would have ultimately led to RNA would have been the ones to propagate themselves. So yeah, basically we don't know, but but yeah, there's a lot of theories out there.
So we know the lightning wasn't necessary then. I always pictured
Well, so
it all there together.
yeah,
and
so the
then-
lightning, it actually, so there's a little experiment I showed up here that had a bunch of apparatus, glassware hooked together, and it had a little lightning bolt going through there. There's actually a famous experiment. It was the one that really kicked off the field of organic chemistry. But what it's the Miller-Urey experiment, if you're familiar with it. But they basically tried to mimic atmospheric conditions on an early Earth and So there was a lot of methane gas and like carbon dioxide and whatever else they pumped into there. But they pumped this all into this system, and then they zapped it with electricity to simulate maybe lightning strikes or something. And then what they did was they collected basically what came out of it and analyzed it and saw. And what they looked and when they looked at it, what they saw was they saw these really simple chemicals that looked a lot like some of the amino acids. So potentially, lightning might have involved in the synthesis of the first amino acids. And then once you have those around, if RNA is around, it can put them together like pop beads into proteins. Yeah.
Could you comment a little on CRISPR and the relationship of that?
Yeah, so CRISPR, I should put that up there with maybe the medical RNA slide, but the CRISPR is all about editing your genes or editing genes in anything. It's actually a fairly simple system. It's developed from the bacterial immune system. So maybe I shouldn't go into too much detail with CRISPR, but I do like it a lot. But CRISPR, it's got two parts to it. You've got an enzyme part, so a protein that can cut DNA, and then you also have a part of it called the guide RNA. So it's an RNA molecule with a very specific sequence that you design. And that guide RNA tells the scissors, the protein, the DNA cutting protein you put in there, where to cut. And so you put both of these parts into a cell, and the RNA basically ends up, in essence, sort of dragging the DNA cutting protein to the exact spot in your genome that you want to cut. And once you cut it there, you can do a couple things. You can basically cut out a gene, so cut it at both sides of the gene, and the gene's gone from the genome. It won't be expressed as a protein. You can also make a cut, and you could insert something different if you wanted to insert a different gene. Or it's so precise, you can actually cut out a single letter, a single nucleotide that might be different. And maybe this is a mutation that's giving you some sort of disease. But you could cut that nucleotide out, put in a correct one, and now that cell has no more disease because it doesn't have that genetic component of the disease anymore. So CRISPR is an awesome system. It has some drawbacks to it, but you can use it pretty effectively, and it's fairly simple to use once you get the handle of it. There's a bit of screening and stuff involved in it, but you can use it in essence to sort of program functionality into single-celled creatures pretty easily. Recently, there's been a lot of stuff in the news about the human CRISPR event where somebody, sort of rogue scientist in China, decided to try CRISPR gene editing in embryos, human embryos, and they were carried to term. So he tried it in 2 girls, and the gist of it is he wanted to introduce a mutation that would make them resistant to HIV-1, which sounds like a good thing. There's a mutation that occurs naturally in a lot of, well, not a lot, but in many places in the world, and it actually arose by selective pressure as well. So it's a mutation that actually makes you also resistant to the Black Plague. So in medieval Europe, Black Plague spread everywhere. Everybody died, but if you didn't have this mutation, you were fine because the disease couldn't affect you. And so they stuck around, they multiplied, and the genes for that mutation spread. And now these individuals, just by mere chance, happen to be also resistant to HIV-1 infection because you happen to need that same protein for HIV-1 to gain access to your cells. And so he was trying to induce that mutation to make these girls resistant to HIV-1. You have to start, it's very hard to get CRISPR into multi-cells in a multi-celled organism, and it only works in the cell that you get it into. So this is why he started with the embryos. At that point, very small, you don't have to worry about getting it into all the cells in the body. You get it into as many cells as you can. And then as those cells divide, then that CRISPR system or that mutation is carried to all the other cells that descend from those cells. And yeah, so it was a great idea. I mean, it's a great idea to think about engineering people resistant to disease, and that's where the technology is heading. Like I said, you could potentially cure all these diseases that are genetically based with this technology, but a lot of ethical questions about how that was carried out and was consent really given, and now we have these potentially human beings that are going to grow up as test subjects, and they're GMOs at this point. They're genetically modified creatures, and if it really does spread to all their cells, including their germline, they're going to pass on that mutation to their children as well. So hopefully it worked, and it's going to be great. Last report I had heard, which is fairly old, but I thought one of the individuals, one of the girls I thought said didn't get the mutation where they were expecting it to be. So one of the issues with CRISPR is sometimes that guide RNA isn't perfect, so you end up with insertions where you don't want it to be, which is why you usually go through a long process of screening it to make sure it works before you do it. But if you don't got a lot of human embryos to screen it on, yeah, you just kind of go for it, I guess. But yeah, so In the future, we probably will have CRISPR-based medicine, but at the current point, we're not looking at engineering human embryos right now. Yeah.
Are you familiar with critical mycelial concentration?
What is it?
Critical mycelial concentration?
Oh, to make the membranes.
Make membranes. Yeah.
I'm not totally. Yeah, I mean, I could get the idea. It's, but yeah, I'm not that familiar with it.
Well,
So.
in Martin Hasnick's demonstration, did he measure, or did the RNA build the membrane around itself?
No,
Or-
he was placing it inside the membranes there.
He was placing.
Yeah,
this
and
on-
in those demonstrations that I show you, the chemistry that he was doing in those droplets wasn't actually RNA. He does have some video of the RNA in the droplets earlier on, but they don't do anything too exciting. They sort of just sit there. The idea would be like, he was trying to show that this chemical definition of life could happen as long as you had chemistry happening in and out of the cell.
It just occurred to me to be interesting if the critical mycelial concentration is when you have enough mycels, membrane layers, that they spontaneously form a cell.
Yeah.
So
Well,
if you could get it to form a cell around itself before it reached the critical mycelial concentration.
Yeah, and
it
I
would
don't know
show
how
that
the
it's
whole setup would have worked. Like, would the RNA have found a pre-existing mycel or a some sort of other membrane and squeeze its way in, or would it have tried to form something around itself in some sort of way? Yeah, my thinking is it probably would have evolved as sort of a on sort of like a film chemistry. So like these things were living presumably in shallow oceans where you had RNA and these things around. If you had some evaporative event that left this this sort of monolayer,
be
right?
concentrated.
Yeah, and if it had enough of these lipids basically laying there in the monolayer and the RNA was on top, and then you had it flood with water, then it might fold up into some sort of ball that happened to contain the RNA in there. But yeah, he may have looked at that in his papers, but I don't know the details of all his work on there.
Does he give a lot of talks or just?
I think he's got a couple on there, and that one's actually fairly old, so he might have something more recent floating around. But yeah, they are pretty interesting. So check it out. Yeah.
Did Luca have, or is it expected that it had primitive organelles, or when did organelles develop?
Yeah, I think it's thought of that this was so some of the organelles like mitochondria and chloroplasts, those were probably endosymbiotic, so it was probably after Luca that you got this. But so you had to be able to do some chemistry to keep yourself alive. You had to be able to make proteins, and you had to be able to get some energy from your environment. So if we look at everything today, one commonality all life forms have is you can do glycolysis in pretty much all the cells. So it's possible that they probably had RNA and DNA and then proteins inside of Luca. No nucleus or anything, but those proteins were probably able to catalyze glycolysis in some way. So they were able to get some energy from the environment, keep them going. Yeah, it would have been presumably more probably anaerobic, because you wouldn't have oxygen around, so glycolysis would have been it. But yeah, organelles, you would have had ribosomes probably to make the proteins eventually, probably not initially, but yeah, no mitochondria, no chloroplasts or anything like that.
And then with ribosomal RNA probably led to ribosomes, or did chicken per se kind of?
Yeah, so I think what would probably happen was it was the ribosomal RNA first wasn't assembled into the ribosomes because the ribosome is made of several different subunits. You just had these random structures of RNA that could just shove amino acids together, and a bunch of them found out, or if they ended up working together, it was more efficient than them being individual, and so they all came together to build the ribosome as the most efficient way of making proteins, and still got passed on.
So the big question would be, for the origin of life would be, and the definition, the newer definition, is what's the basic unit of a chemical that could replicate itself?
Yeah, you'd want to find the minimum amount of a chemical that could replicate itself with some sort of, so you'd have to have some retention of some genetic information. So, I mean, you could say a nucleotide that was, or an RNA strand that was maybe 2 nucleotides long, maybe, because then you have some sort of sequence that would contribute to it. But yeah, that would be the question. So, how simple can you get as far as RNA goes to still fulfill that definition?
So that's what you should be looking for in astrochemistry, where you're finding new chemicals that you haven't had before, or
Yeah, well, I mean, I think like with the polyaromatic hydrocarbons, if people were to find those on some sort of planet, that would get biologists pretty excited as well, because then at least you have potential there for them to assemble and for life to evolve from that point. Yeah.
But then sorry,
Uh,
but that wouldn't leave behind any skeletons. So you
yeah, that's the hard part.
wouldn't know if they're still there on
They don't leave
Mars.
fossils or anything
Yeah.
behind, so you have to find them, there in real life. So that's the tricky part of it. Yeah, question.
Yeah, so we have the benefit of 4.5 billion years of looking back at all this through science. If you put yourself before life, what's the imperative towards anything? What's the imperative towards complexity? What's the scientific? There's nothing that explains complexity.
Yeah, so basically complexity is arising through competition. So, you get RNA competing for nucleotides, and animals competing for, resources. So it's really just, the environment is changing over time as well. So
You're
Earth,
using an organic word, you're using a word competition.
That's selection.
yeah,
These
so
are all words that don't mean anything to the
right, like
chemical beforehand.
Yeah, exactly. So this would imply, the RNA doesn't know that it's competing with anybody. We're saying competition because that's what it looks like from our perspective. But they're trying to do a job, and somebody else is trying to do the same job, and there's only a limited amount of resources. But yeah, what it would suggest is that what we're doing is sort of trying to hit a moving target with life. So life it's just sort of evolution is this inherent property in the universe, it would appear, or at least on Earth. And so evolution, if it can affect everything, even chemicals, but even those chemicals are trying to be the best fit for the environment that they're in. Whether they want to or not is not the question. It's just the most successful ones, the best fit for that at any given moment are the ones that are going to thrive.
But inherent's not a scientific principle, that's just.
Right, yeah, I don't know how evolution is encoded in there, but yeah, looking at this, if chemicals can evolve, then yeah, there's something to say that evolution can extend beyond biological form, so why not apply it to chemicals, I guess. But I don't know what the actual, explanation for why that is will be there.
I wouldn't worry though, entropy is maintained.
Well, if you have more complexity, then isn't that counter to entropy?
No.
Yeah.
I think the more you analyze it, the more you get to philosophy.
Yeah, at some point,
Philosophy
we
is,
started with a little bit of biology and we moved into chemistry, and then at some point where it's just purely conjecture.
I mean, it all boils down to physics and math,
Mhm.
and it's all based on laws.
Yeah, and if you look at the evolutionary tree, complexity is increasing. We get more life forms, more adaptations showing up, and I think that's in some way, that's where we're going to get the best selection, right? So we got the best stuff to start with, but as things go on, the Earth's environment is changing, more and more quickly, and we're going to see more and more of those adaptations being selected for and against. And so complexity is a great thing to have because it keeps you around. So genetic diversity in a species is something awesome because everyone won't get wiped out by the same disease. If a disease kills you, I might be alive because my genes are slightly different. So genetic diversity in a population is great for the survival of that population. Genetic diversity in a species, for the same reason it's great. So genetic diversity, among all the life forms on Earth, the more different diversity we have on planet Earth, the better it is for Earth as a whole, because chances are, no matter how the environment changes, there's going to be something that can thrive and pass on some genes.
I think one of the challenges is trying to avoid the anthropomorphizing the process.
Right.
Saying that this thing is trying to accomplish something.
Yeah.
So to come out, but it's difficult to come up with
There's no
another
goal, it's just sort of
semantic to describe what is happening without making it look as if there is some intention on the part of the things that are doing what they're doing.
Right, yeah, and I don't mean to imply that anything is the chemicals aren't thinking, they don't have a directive of their own. They're just doing their thing, and that thing happens to be chemistry, and that chemistry happens to require chemicals which are only available in limited quantities. So you have selection happening.
Thanks again for the.
Cool, thank you.